We're going to pick up where we left off with the last lecture, and we're going to talk about monohybrid crosses or one-trait crosses. So one-trait crosses, a homozygous man with a widow's peak, and it's a one-trait or monohybrid because we're just looking at widow's peak, nothing else, reproduces with a woman that has a straight hairline. So what are the possibilities for the parents? Well, if the man's homozygous, that means that he has to have two identical alleles to pass on to his offspring. Same thing with the woman. She has a straight hairline. She's homozygous recessive, and he is homozygous dominant. It wasn't important for them to put homozygous recessive with the woman because it says she has a straight hairline. The only option for a straight hairline, if we look back at this chart from yesterday, the only possibility would be little w. But it was important for them to tell us that the man was homozygous because that told us the genotype that he had. Otherwise, we would wonder if he has big W, or if he's heterozygous, big W, little w. So this one is a really simple concept to grasp because the man can only give a big W for each one of his sperm, and the female, on the other hand, can only give a little w in each one of her eggs. So the only possibility for their offspring then is to have a widow's peak and to be heterozygous, in this case, for widow's peak, big W, little w. So every one of their children should have a widow's peak. Look at another example. All right, here's a one-trait cross, and this is where the Punnett square comes in. It isn't as easy to do, so we're, as the first one was, so we're going to look at using the Punnett square to help us figure out statistics when it comes to this. So a one-trait cross or monohybrid, I'm going to go ahead and write that down here because I think that's important for us to connect with one-trait cross as a monohybrid cross. So two individuals are both big W, little w. So this is information that's already given. So here's the dad and here's the mom. And we're going to use a Punnett square to determine what the possibilities for their offspring are. Because they're not both homozygous dominant, we can't automatically tell what it is. So if this is what the male is, each one of his sperm are either going to give a big W or they're going to give a little w to the offspring, depending on segregation. The female is the same way. Her eggs are either going to give her offspring a big W or they're going to give her offspring a little w. The way that we fill in this Punnett square is like filling in a multiplication table. So we just go what's in this box and bring down what's in this box. So this box is big W, big W. This box, this way and this way. And I always just get in the habit of putting, if you have a capital letter, put it first in your box. It's going to make it so much easier to look at and find the information you need. So big W, little w. This box is going to be a combination of here and here. So we have big W, little w. And this bottom box is going to be here and here. So little w, little w. And when you do a Punnett square, there's some things that you have to do every single time. You have to tell me the phenotype and the genotype, or the phenotypic ratio and genotypic ratio. Let's look at the phenotypic ratio to start with. This individual has at least one copy of the dominant gene. So this person will have a widow's peak, as will this individual and this individual, because there's at least one copy of the dominant gene. This one, however, has no copy of the dominant gene, so it's going to be recessive, homozygous recessive, and have a straight hairline. So the ratio. Several different ways you could write this. You could say that it's 75% widow's peak, 25% straight hairline. You could say it's in the ratio of 3 to 1. It's up to you how you write it, but you have to always tell me what it is. For example, if I want to write 3 to 1, I say 3 to 1. Widow's peak, I have to say 3 widow's peak. I can't just say 3 to 1. The phenotypic ratio is 3 to 1. I have to say what it is, 3 of what to 1 of what. Or like I said, we could say that 75% widow's peak and 25% chance of having a straight hairline. So what this square is telling us is if this individual has 4 children, statistically 3 out of the 4 would have a widow's peak. Now the genotypic ratio is the information that you get from the genetics of it. So we look at the first one and we have big W, big W. We have big W, little w. There are 2 of those. There's one big W, big W. There are 2 big W, little w's. And there is 1 little w, little w. So this is what's expected when I ask for the genotypic ratio. it would be 1 to 2 to 1. And you can't just say 1 to 2 to 1. Again, you have to connect it to what? 1 watt to 2 watt to 1 watt. So when you do a Punnett square, you do the Punnett square, you give the phenotypic ratio, and you give the genotypic ratio as well every single time. One trait cross and probability. The chance of two or more independent events occurring together is the product of their chance occurring separately. So in the cross, big W little w by big W little w is what is the chance of obtaining either a big W or little w from a parent. The chance of a big W is 1/2. The chance of a little w is 1/2. So if we look at the probability of having these genotypes, this is what we would come up with. And this should be something really familiar because you've done Algebra 2 information that's going to talk about probability and chance, and that's what this is showing you, the math behind what we're doing with the Punnett square. So the chance of big W is 1/4. Chance of big W, little w, again, is 1/4. And then here they switch it, little w, big W. In our world, those are the same thing. These two are the same. And that would be 1/4. So we add those together and have 1/2 of a chance. And then big little w is 1/4 of a chance. So let's go back and write that on there. So you notice that the chance of big W is 1/4. And this is our genotype ratio. This is 1/2. And remember, our ratio is 1 to 2 to 1. And that's what you see statistically when you look at doing the probability. So what happens if you have an individual who has a dominant trait showing? Do we know if that individual is homozygous dominant or heterozygous dominant? We don't know just by looking at them at their physical appearance because those have the same genetic makeup. So we do a test cross in order to determine whether that individual is homozygous dominant or heterozygous dominant. The way that we do a test cross is to cross it with a recessive individual because we know the genotype. If we see, for example, if we go back to the hairline, we see an individual with a straight hairline, we know automatically they're little w. So we take an individual that we know that is recessive for that, and we do a cross with them to find out what the possible offspring are. Look at the offspring and compare them to the Punnett squares. All right, so here's an example. We have one parent that is obviously has a widow's peak and one parent that does not, but we don't know if the parent is big W little w or big W. Well, if we look at the possibility of their offspring, what they would have, we look at this, we have a possibility of 2 out of 4 having a widow's peak and 2 out of 4 having a straight hairline. So if these two individuals had children, we would expect to see a ratio of 1 to 1 as far as the number of children have the straight hairline and the ones that have the widow's peak. So these pictures here are two test cross right next to each other. This one is a parent. What would happen if the parent's homozygous dominant and we cross it with a parent that is homozygous recessive? Then the Punnett square tells us we should have all 100%, all of the children, all the offspring will have a widow's peak. On the other hand, this is the square we just looked at on the previous slide. If the individual is heterozygous dominant for the trait, we do a test cross, and half the children should have a straight hairline and half the children should have a widow's peak. So that's a test cross. We take an individual, we don't know exactly what the genotype is because it's dominant, and we cross it with a homozygous recessive individual, and we look at what their offspring would be. Now, this isn't something you would do, of course, with humans necessarily. We do this more with animals and plants. Practice problems. Both a man and a woman are heterozygous for freckles. Freckles are dominant over not freckles. So what's the chance the child will have freckles? And you have to show the Punnett square, give the phenotype possibilities of the offspring, and the genotype, because that's what you have to do. This is what I expect to see when you have a problem like that. So the first thing you're going to do is you're going to write down what you have for the male and the female, or the two parents. So they're both heterozygous for freckles. That means that they both have one big F and one little f. We're going to show that here. Then we're going to do a Punnett square, and this parent can, and I always just out of habit This one I always put on the left-hand side. This parent is either going to give a big F or a little f to their offspring. Kind of crowded on here, but this parent I always put up here at the top. This parent is either going to give a big F or a little f. We fill in the boxes. And like I said, this is exactly what I would expect to see. If you have a problem like this, this is what I have to see. So I do the phenotype ratio first. So I write down phenotype or just pheno. So I have to identify what they look like. So we know that one big F will give you freckles. So we have freckles, freckles, freckles. So we have freckles, abbreviated 3, and no freckles 1. You can look at that as 75% to 25%. You can do 3/4 to 1/4. It doesn't really make any difference. Genotype is what the genetic makeup is. So genotype, we have 3 different genotypes. We have one that's big F, big F. We have 2 that are big F, little f. And we have 1 that is little f, little f. So this, when you're asked to do a Punnett square, this is what I'm looking for. The question was, what's the chance the child will have freckles? And here's your answer. Chance of freckles is 75%. Look at the last slide for this lecture. Both you and your sister have attached earlobes, but your parents have unattached earlobes. Unattached earlobes are dominant over attached. What are the genotypes of your parents? So let's stop and think about this for a minute. So you and your brother have attached. Unattached or dominant. So you and your brother have attached, and that means that you must be both recessive. So you have unattached. It says, what are the genotypes of your parents? Show the Punnett square and give the phenotype possibilities of the offspring. Well, obviously your parents, if they both have unattached earlobes, like it says right here, that means that they have to have at least one copy of a dominant gene. On the other hand, it's pretty obvious that they can't have 2 copies of this dominant gene. I'll show you why in just a second. But if you and your sibling are homozygous recessive, you had to get one of these from mom and one of them from dad. So they have to be heterozygous in order to produce those offspring. So if I were to do the Punnett square, which you would be expected to do in this answer, and give the phenotype and genotype ratios, it's going to look exactly like the one that we did on the slide before this, but you're going to use E's instead of F's because you're referring to the earlobe instead of the freckles. So practice on the monohybrid crosses. It's not that big of a deal. It's going to set us up for a dihybrid cross and get us into codominant and incomplete dominance as well as dominant.